Hi, this is Miss Michael, and I'm going to go over the process of meiosis, which is a process that occurs in sexual reproduction of organisms. Meiosis is a process that shows how we get variation of traits. If you look at this family in this picture, you can see that the mother and father, there are two parents, and the fact that there are two parents is going to be a contributing factor to creating variation, but also the process of meiosis that occurs in males and in females that brings more variation to the offspring. So you may wonder why you don't look exactly like your siblings, or maybe you have a brother or a sister and they might look a little bit like you or they might not look like you at all. That is because meiosis is has been a random way for sexual reproduction to produce genetically different yet similar offspring. Um, Case in point, the family in the video. So if we look at meiosis, you have the male and the male and the female each contribute half of the amount of chromosomes. Okay. In this process, the male has a total of 46 chromosomes and the female has a total of 46 chromosomes. Okay. Cells that have a full set of chromosomes are called diploid cells. So the female and male are going to produce gametes by means of meiosis. And those gametes are produced by what we call a germ cell. So the sperm is going to be a haploid cell, and the egg is also going to be a haploid cell. They are called haploid cells because they have half the amount of the full set of chromosomes. The full set is 46. The half of the set is 23. So 23 chromosomes are coming from the sperm or male gamete or male haploid cell, and the other 23 chromosomes are coming from the female haploid cell or the egg cell. Now, meiosis, as I said, results from what is called a germ cell. Okay. A germ cell is going to come from the gonads of male or female. And so in the process of meiosis, you have the cell that is dividing twice. So in a germ cell, you have meiosis one, which is one division, and then meiosis two, which is the second division, which is different from the process we learned in mitosis. Okay, meiosis one and meiosis two. Mitosis is a process that occurs in somatic cells, cells that have a full set of chromosomes. Okay, and in this scenario, you have mitosis producing the two daughter cells. They have 2N, which represents a full set of chromosomes, a diploid cell, and meiosis 1 and 2 are producing haploid cells. These haploid cells have half the amount of an original set of chromosomes. So far, the difference between meiosis and mitosis is meiosis produces 4 haploid cells that have half the amount of chromosomes of the original germ cell, and mitosis produces 2 diploid cells. Now, the purpose of mitosis is for growth and repair of somatic cells. The purpose of meiosis is to produce sex cells so that a sexual organism can reproduce. So, before meiosis occurs, the cell is going to go through G1S phase and the G2 phase. Okay, it still goes through interphase where DNA is replicated in the synthesis phase. Okay, where you have here you have those sister chromatids. All right, and then as the cell moves into prophase, you're going to have the centrioles develop, and the centrioles are going to move to opposite sides of the cell. These are your centrioles and spindle fibers that are forming. So, in the first phase of meiosis, you have a couple of things that are really important happening that are going to create genetic diversity. That first phase is known as prophase one. Okay. You have homologous chromosomes. Okay. Homologous chromosomes are simply chromosomes that are the same size and have similar genes. Not the same genes, but similar genes. These homologous chromosomes are going to pair up in a process called synapsis. All of this happens in prophase one. It's the first phase of meiosis. Okay. So the second thing that is going to cause genetic diversity in meiosis, or in this gamete production, is something called crossing over. It's where the homologous chromosomes that are paired up have come together, and they will shuffle traits between the chromatids. And you can see this in this animation, that you have you have traits shuffling in that process of crossing over. So synapsis and crossing over in prophase one are going to be two things that are going to create genetic diversity. Okay. So as the germ cell moves through prophase one, okay, and those chromosomes are paired up, they've shuffled traits. Okay, they're going to attach to some spindle fibers, and these are the fibers that are shown here in yellow or in orange. And so here it shows a graphic of chromosomes in meiosis and chromosomes in mitosis. You can see in meiosis there's been a shuffling of traits, and so those chromosomes are attaching to spindle fibers. And the next phase of meiosis one is going to be metaphase one. In metaphase one, they are going to line up in the middle. They call that the metaphase plate. Okay. And they meet up randomly. So that's another thing that causes some randomization. Then we go into anaphase one, and you can see in anaphase one that those chromosomes moving away from that center, they don't look alike. There have been a shuffling of traits. Okay. So then you have your first split. You have the cleavage flow occurring, and we head into metaphase two with two of these cells where they are going to continue uh, to create those gametes. Okay. Prophase two, you have two cells that are going through that process where the centrioles are moving to the other side, prophase two. And then you have your chromosomes that are attaching to the spindle fibers. And again, in metaphase two, you can see that the chromosomes are meeting up in the middle of two different cells. You have two cells where chromosomes are meeting up in the middle. Then you have the split. You have sister chromatids. Okay, you see that the chromosomes have split in anaphase two, and chromatids are moving away from the center. Now, the chromatids are part of the chromosome that were attached. Okay, sister chromatids are attached by a centromere in a chromosome. So in anaphase two, they move away. And again, in telophase two, you have the nuclear membrane beginning to develop, followed by cytokinesis two, where you are left with four genetically similar yet different gametes. Okay. Meiosis produces four haploid gametes. Then you have fertilization. You have the sperm and egg cell that join together to produce a full human. Half of those chromosomes coming from the male gamete, the sperm, the haploid male, and then the other half coming from the female gamete, which is the egg. That compares mitosis and meiosis. so you can kind of take some notes. Both are divisions of a cell nucleus. Mitosis produces somatic cells, whereas meiosis produces gametes. You have in mitosis only one division occurring, and in meiosis you have two divisions occurring. Mitosis produces two diploid daughter cells. Meiosis produces four haploid daughter cells. Mitosis produces clones. These are genetically identical cells. Meiosis is going to produce genetically different cells, less than 1 in a million alike. Okay, the purpose for mitosis is for growth and repair, and the purpose for meiosis is for reproduction. Okay, and there's no synapsis and crossing over in mitosis. You don't have that shuffling of traits in mitosis like you do meiosis.